I'd like to introduce Dr. Hubert Lim with Neuromod.
Hello, everyone. Thank you for sticking around for this long 30-minute talk for a 3-hour session. This is a bit different, a little bit more on the geeky side, a lot of data I'm going to present, so hopefully it all can be absorbed well. So I'm a professor at the University of Minnesota in the Department of Otolaryngology and also in biomedical engineering. I do have some disclaimers here. Even though it's research that I have done at University of Minnesota, I am part of a company that some of you may know already, Neuromod Devices. I'm serving as a consultant there as their chief scientific officer. It's been managed through the conflict of interest, so please take that disclaimer. Also part of 2 other companies that aren't relevant for today. So the way I want to organize this talk is rather than talk just about linear data, I want to give you a perspective because I've been working on bimodal neuromodulation for about 15 years now. It seems like a long time, but there's a lot of research that has built up towards bimodal neuromodulation. So I want to give you the broad view, and then I want to go into the results of the clinical trials I was involved with linear, and then what's more important for me is real-world data as that transitions from clinical trials to the clinical practice. And it starts to get more and more independent of data because other people are doing those clinical evaluations and treatments. And I want to acknowledge Dr. Thomas Lennertz in Germany, which is one of the first clinical sites in Hanover in Europe when it first came out in Europe. And then in New York, Dr. Craig Casper was generous enough to share this. He just presented his data last week in Boston. So I think it's a lot of exciting data to share today. I'm sure all of you know what tinnitus is. I show this slide because for me, when I talk to a lot of my colleagues, I'm in the neurotechnology realm. And my colleagues work on diabetes, they work on pain, they work on epilepsy, Parkinson's, and when I show them the 10 to 15%, and this is not all the people with tinnitus, these are individuals that are bothered by tinnitus. They're actually surprised, and they hadn't heard about tinnitus, and the funding sources coming in are not anywhere close to what my colleagues get in the other fields. So it is a hidden condition. I do feel it's gotten better. But I think this is something where, as we continue to push the federal funding agencies and public awareness, we're going to be able to have more and more research and opportunities and treatment paradigms. So I think this is important for me, just understanding how big of a health issue it really is. There's many different treatment options, that all of you in your clinics try. I'm going to focus on 2 because it sets the stage for why bimodal neuromodulation. Hearing therapies are quite easy to use. They're accessible. A lot of people feel comfortable trying them out. Now, I know in the clinical guidelines it's not recommended per se specifically for tinnitus, but I think we all can agree that it can be effective for many individuals. I do appreciate Sarah Theodoff and Jim Henry at VA. They published a nice paper to reassess the guidelines, but I think that's something to keep in mind. Sound stimulation can work. We all know sometimes we need a bit more effect size, and the long-term effects may not be there for many tinnitus patients, right? On the other side, there's cognitive behavioral therapy, psychotherapy approaches. These are in the clinical guidelines. They do work well, and they can have long-term outcomes, but accessibility is not as broad as what you would see with sound therapies, though things I would argue are changing in the brain on a long-term scale. I will say that internet-based CBT, counseling approaches online are starting to come to fruition. Some of you may have been using them, so I think that's an encouraging realm as well. So how do we get then the best of both worlds? That's what we're trying to do here. We want to have something that's a simpler device that you can implement in your clinic, provide patients access to, but at the same time driving greater, longer changes in the brain. And we have to step back a little bit, and this is going to be very, very simplified, but the idea is that tinnitus is ultimately being coded in the brain. Whatever the cause may be, but I would actually argue that most tinnitus patients have some form of hearing loss, whether it's hidden hearing loss or suprathreshold deficits, but that lack of input then is going to reduce the input that makes it up to the auditory cortex, and then the brain tries to compensate, which is great, but then it overcompensates, right, for some individuals. And as a result, you have tinnitus. Now, we know that sound can interact with those networks. The question, though, is in a simplified way, can we somehow amplify the effects that are happening with the sound? Because we know sound can shape the brain, sound can interact with the brain. We've heard a few talks, long-term benefits of sound. But imagine if you had kind of like a super sound therapy where there's a reinforcer that's driving whatever the sound effect is doing. And you can do that with different non-auditory inputs. And then as a result, the idea is that you can be able to reduce tinnitus. So this is where it gets a bit geeky. Please bear with me, but I hopefully you'll be able to absorb it and appreciate it. There's been a lot of research with bimodal neuromodulation, repaired plasticity. Over 100 years of research. There's behavioral evidence. There's physiological evidence. 40, 50 years of physiological evidence. And where I come into the picture, and a few other groups, is the third bullet point, where we're just trying to figure out how to tap into that plasticity network using non-invasive approaches and bimodal stimulation of the body and sound. So this is the simplest way to start. We all know about Pavlov's dog. Poor little dog there wants to eat the food and doesn't care about the bell. But if you pair the bell with the food, then this meaningless sound becomes meaningful to the animal. And it's pretty potent. For this kind of food situation, it only takes a few trials. And even if you read the papers, they talk about the individual bringing the food up the staircase and the sound of the stepping on the staircase causing the dog to salivate. So it's pretty potent. Now, I don't think you want to serve food at your clinic and treat tinnitus this way. So there's got to be other ways that we could try to tap into this reinforcer. Now, you might be wondering, what does this dog, Pavlovian's condition, have to do with tinnitus? But just to set this in your mind as I'm going through the next slides, when you think about tinnitus, there's two ways you could try to tackle it. One is shut down the tinnitus neurons or the sound. That is possible. I've done some more deep brain implantable techniques where we can target some of those regions, but there's another way to do it, and it's more distracting your brain away from tinnitus. And so you can think about that bell being some other sound other than tinnitus, and you could play that sound. For example, if you have tinnitus, let's say at 4 kilohertz, you could play tinnitus at 8 kilohertz or 6 kilohertz, and you provide a reinforcer. Your brain then is going to start to become more sensitive to that and shift its attention to that new stimulus. Then you provide 2 kilohertz and 1 kilohertz, and you keep jumbling it around. Now, you don't want to do the same stimulus over and over again, because I would hypothesize that you would potentially induce tinnitus, right? And we all have cases of that in our daily life where we've heard something over and over again, maybe a concert, and then you go home and you swear that you're hearing that sound or that music, right? So then now, how do we actually implement that to try to shift our brain's sensitivity and coding away from the tinnitus and towards these other sounds? And you can leverage the beautiful tonotopic organization of the auditory system that you're all familiar with. Now, when I was a PhD student at University of Michigan, I thought this fundamental organization was fundamental, that it was stable and fixed, and it was serving as the anchor of your auditory system. I didn't think that you could drastically shift this fundamental organization in different ways. And so this is why this paper, when I came across, there are multiple papers, but it led to a review paper by the late Dr. Weinberger. He was a professor at UC Irvine. He ran these seminal experiments, many of them, but this is one example. It's one of my favorite figures, is he took guinea pigs. He basically then recorded from a cell in the auditory cortex and characterized what sound or what frequencies that cell is sensitive to. Then he stimulated the body. This is more intense stimuli, suprathreshold stimuli, paired it with a tone that that neuron was not sensitive to before, in this case 2.5 kilohertz, and then change that cell's identity, right, to become sensitive to that frequency. So if you look here, you have the x-axis frequency. It's like a reverse audiogram. Y-axis is spiking of that cell, for example. Green curve shows you that when you play 100 hertz to the guinea pig's ears, 200 hertz, doesn't really care, 500 hertz, you play about 1 kilohertz, that cell lights up, right, super active. And then you play 5 kilohertz, 10 kilohertz, doesn't really care. So we say that cell's tuned to about 1 kilohertz. This is the part that I was astonished because I didn't think you could actually do this until I came across these studies. But then you do only 30 trials of body stim, 2.5 kilohertz paired, and then look what happens. Few things to notice. The orange curve, everything's a little bit elevated. Okay, you got the cell going, but it's not as sensitive anymore to that 1 kilohertz as much. And it is super sensitive to the 2.5 kilohertz, right? So this already shows you the power of being able to use paired stimulation to shift the identity of cells. So going back to what does this have to do with tinnitus? Well, if there's tinnitus cells and they're stuck on coding the tinnitus, and you start blasting these cells with paired stimulation and confusing those cells basically and shifting their identities, there's no way that that cell can code the same way it was for that tinnitus percept. And moreover, you're making more cells more sensitive to other sounds. Okay? Homeostasis is important. Your brain can't be sensitive to everything, so it's going to start to dilute its attention and coding to different sounds as it's becoming more sensitive to other sounds in this extreme way, right? Now, that was more of a strong stimulus. They use it for fear conditioning, but there's a more subtle stimuli that's possible, right? This was another seminal paper by Dr. Michael Kilgard's group at UT Dallas. I love this paper. Basically took implants, vagus nerve implants, and they electrically stimulated the vagus nerve, which is a strong reinforcer. I won't get to the details there, but they basically did a similar experiment. They recorded this time from many cells in the auditory cortex, characterized the tuning of those cells, and determined the percentage of cells that are tuned to 1 kilohertz, 2 kilohertz, 6 kilohertz, 8 kilohertz. And so on the x-axis on the left plot, you see the frequencies. The y-axis, you see the dark black curve with the black circles. 14%, 13% are sensitive to 1 to 2 kilohertz. 2 to 4, there's about 14%, so forth. It's not really flat, but it's roughly even. And then they do stimulation of vagus nerve stim with 9 kilohertz, and like you saw on the other plot for single neuron, population of neurons are shifting their identities, right? Now it takes longer. In this case, it took 6,000 trials over 2 to 3 weeks, right? But it's possible. And again, if you do look at the homeostasis, things have to rebalance itself, right? You could do it with pretty much any frequency. They did 19 kilohertz, but you could do all sorts of different frequencies and then just kind of almost have a tool to shape and shift things around, right? So this is where my lab came in about 2009 when I got to Minnesota, where I was trying to figure out how could we do this non-invasively. And we know that there's lots of body projections to the auditory system. And so you could tap into any of those body regions and pair it with sound. Now, we did do some human studies. It just takes a long time to go through all those parameters. So we went to our fluffy, friendly friend, the guinea pig, and we could stimulate a lot of different body regions, play lots of sounds, record in different brain regions, the midbrain, the auditory cortex, and we could get a sense of which body regions, what patterns are greatly driving the plasticity effects, right, that we want. I don't have time to go into it. You can read the paper. It was published in 2015. The two take-home points: bimodal stim drove stronger changes than sound therapy alone or sound stimulation alone. Other groups have shown this effect. Dr. Susan Shore at Michigan, also in her animal work, and in humans showed this effect, and I'll get to some human data. The interesting thing was we found two body regions that drove the strongest effects. Electrical ear stim and sound made sense to me, considering the ears involved, and connected some way to hearing. Tongue stimulation was surprising, and I, at the time, knew of the company Neuromod devices. They had started in 2010. I will be honest, they're actually a competitor at the time because I was trying to do my own device. But I thought this was interesting, and we published it, and that's how Neuromod devices got in contact with me to try to collaborate, to try to push this along. And one of the things that I found fascinating was they had some investment money to run large-scale clinical trials that I could never do in my lab, and we can do 500, 700 participant studies. And so that was what drew me to working with them to really figure out how to make it work. And electrical ear stim I've continued in my lab. So this then starts the story of Neuromod and Linear. Based on all that, I'm still working on the ear stim, But this has brought us along a pathway for the last, 6 years now, trying to figure out how to make tongue stim and sound work for treating tinnitus. So this is the device. If you haven't seen it yet, it has a controller, a tongue tip that goes into the mouth, providing the electrical stimulation, and the headphones are Bluetooth that allow you then to do the combined stimulation. Now, all the publications, we're trying to be as transparent as possible. We published protocol papers and then applied for, did the study, and then also present the results. We've run 3 clinical trials. The original goal of these trials, the first 2, because it's such a huge parameter space, was to do a funneling approach. We wanted to do lots of different stim settings in bundled ways because we didn't want to do 10 patients or 10 participants, one condition, 10 participants, one condition, do 50 conditions. We bundled them with many different settings and did 3 groups of about 100 participants each. Based on 10A1 findings, we found out what were some key features. We narrowed that in, unbundled some of them, and did 10A2. And then in 10A3, we discovered, and I'll get to the data, the key findings are that tone stimulation and tongue stimulation, those are the key factors, and some of the background features that we had and other kinds of synchrony patterns weren't as critical. And then working with FDA was really great to work with. I just want to emphasize, if you are running studies, just approaching FDA early, and they worked collaboratively with us to actually design our study to make a controlled a clinical trial, confirmatory clinical trial, and that's how we got FDA approval in March of 2023. So this is all available, the details, and I'll go through briefly these different studies. But here, we designed a 3-arm study, and many people asked us, geez, it was like 4 years ago, 3 years ago, where's the control? This device had already been out for a certain amount of time, commercial, and then they brought it back in to do the clinical trials. It has super thresholds. So if you have in your clinics already or eventually will, you can feel the tongue stim, you can hear the sound, so blinding becomes a challenge. So we mimicked our study after drug studies where they have active doses. You have different dosing parameters. It's called an active control. So we have 3 bimodal stim settings, and so the first 2 are more like stronger stim settings, and then the third one is like a lower dose setting. We had them go for 12 weeks. We took the device back after 12 weeks, and then we tracked them for 1 year. And that was appealing to me that the company and investors were willing to do that, to really look at large-scale studies and long-term data. And we had 2 outcome measures, THI, TFI, which you're all familiar with, and some of the inclusion-exclusion criteria there. These are the stim settings. Ultimately, PS1 is what's offered mainly in the U.S. Some of the other ones are offered in Europe. But PS1 is, think of it more of the synchronized stim. The tongue is stimulated on a given location, 32 electrodes, one of those, 16 split in 2, but one of the locations, and then you play a certain tone, and then you play a different tone and stimulates a different location on that paddle. The frequencies are quite broad, and there's some background noise, and we call that the synchronized stimulation. Because we know that there's different delay lines and different connections, we thought that delay parameters would be important, and we wanted to try to activate as many cells as possible. So we added a lot of delay patterns into the second condition, as well as adjusting some of the synchrony of the tongue stimulation. And the third one was our most orthogonal setting, lower frequencies, longer delays, but also had background noise. So this is a snapshot first, and I'll show you more of the data. And by the way, the paper has all of the data for each participant and how they did throughout the study, so you can look at the paper when you have more time. This plot here, it's just summarizing all the individuals that came to every visit. You could see at y-axis is improvement in THI. TFI, we also saw consistent results, so I'm just presenting THI. Interestingly, what we found is during the treatment phase, all 3 arms led to comparable, I would say statistically, improvement by 6 weeks. And then there was a plateau effect, and there was some differences that were trending, but I would be cautious there because it was post-hoc analysis. But the arm 1, our synchronous, was more flat. The arm 3, which was most orthogonal, was approaching baseline, and then arm 2, which was a hybrid, was somewhere in the middle. So this was encouraging that bimodal treatment can drive effects, 7 points being the clinically important difference, but it was interesting to see this plateau effect and some lack of difference between the arms. Here I'm just showing you the actual data of each participant. The circles are each participant. X-axis is before they got the treatment. Y-axis is after they got the treatment. And you can see anything below the diagonal line is an improvement in their tinnitus score. And you can see just some examples of people having maybe 30, 40, 50, and then being able to drop down below 10. Some, having 7 points, and others just hovering around that line. But improving at least some THI score was 86%. So this brought us now to 10A2. We learned a lot from that first study, and then the question was, what do we do next to further refine the stimulation setting and learn from it? And so the rationale was two things. One was, and this was a push from me, and long story short, I got it wrong, but in my animal research, we used a lot of noise inputs. And to me, I thought that would be a stronger driver of therapeutic outcome. So I personally pushed that the reason why all three arms were benefiting was because it was the noise. So we wanted to get rid of the noise component and see if that had an effect compared to the ones with noise. So that was one goal. And the other thing was, could we break through that plateau effect? Because we know from plasticity studies that things start to adapt. So you got to keep things fresh in the brain a bit, and then you can potentially drive further plasticity. So the way it was designed is we had 4 arms, because again, we want to keep the N sample size large for each arm. So we had 2 arms that are presenting the original PS1 synchronized, and then we had PS6 there that had no background noise, right? And a few other tweaks in that setting. But then we can compare those 2 arms, and that was what we powered the study for. And then we had 2 other arms that were exploratory arms that allowed us to look at different things. And midway through, we changed the stimulation setting so that they couldn't perceive it, and we wanted to see what would happen. Could we further drive improvements? So this is a summary, and again, the paper's available online. What we found was there was a slight improvement with PS1 compared to the no background case, but my hypothesis was wrong. Basically, both statistically were equivalent, that support that sound really is the tones combined with tongue stim that are critical, those 2 components, right? What was encouraging is when you keep giving treatment and changing things around, we're able to increase another 50% of performance, right? And this was encouraging because the individuals couldn't tell that their stimulation settings were changing because it's imperceptible. We changed the delays and some of the tone patterns. In terms of just, THI scores are a little bit tricky sometimes, but, what does it really mean for the participants? You can assess that by looking at compliance, how much are they using the device, and also how they perceive they did. So we had 84% compliant. We recommend it's used 1 hour a day, every day, but we, based on previous studies, required at least 36 hours over 12 weeks, so 3 hours on average per week. So that's what the minimum compliance is. We had 84%. We just asked them, did you benefit from this treatment? 70% said yes. This was interesting. 88% said that they would recommend it, meaning some individuals who didn't benefit were actually willing to recommend, meaning that it was acceptable to them, right? And just to bring this full circle with the whole point of that original concept, or rationale, we're trying to create something that's easier to use, accessible, can have quick effects, like you see with some of the sound therapy approaches, or at least accessible effects, but long-term effects that you potentially could see with cognitive behavioral therapy. And I think we're getting there. I want to be cautious. We're comparing data across studies, which you're not really supposed to do, but I want to give you an order of magnitude. So I won't say one is better than the other, but the key thing is the time course. With CBT and usual care, psychotherapy, it takes a bit longer, whereas with bimodal stim, we can really drive that down. And you could see there that, over time you could get additional effects by changing stimulation settings or continuing treatment. Okay, so then this brings us to our 10A3 that led to FDA approval. What was interesting was that we had this one arm, and remember our whole goal was to take our study, bundle settings, and then unbundle things and try to figure out, hone in on what features are driving things. So I designed a study where the last arm, we then pieced out the tones and the tongue, which is pretty obvious to do next, right? We had the tone and the tongue. Now let's pull those part out. And we had the sound given, and then we followed it by adding the tongue component, and we showed quite strong results, positive results in that exploratory study. We tried to get FDA approval with that, but FDA said you need more than 15 participants, so we had to go during COVID and actually during COVID pandemic, run a large-scale study, which was not fun, but we managed to do it. And that's where everybody was asking, when is it coming to the U.S.? But we had to go back, but they appreciated that data. So we designed our study then, just like that data, to show that adding the tongue component drives further benefit than sound therapy alone. It's all up on clinicaltrials.gov. We're actually in the process. It's in review for publication, so please be patient. We'll get it out soon. But these are the top-line findings. 79% experienced a clinically significant improvement across the study. 82% were compliant, so very consistent with our 10A1 and 10A2. 88.6 said they would recommend. The key thing that we got FDA approval for some of you don't know, is that we actually showed that those with moderate or more severe tinnitus based on THI, so that's a 38 point or above, had quite significantly more improvement than those less than. I'm going to show you some real-world data that supports that. It was safe, no serious adverse events, and we also had real-world data at the time that we combined with our submission, and that's what all led to FDA approval in March. So this is the part that I'm excited about because it's real-world data. Clinical trials are controlled. We do animal work. It's great to see human data in clinical trials, but then we got to get the real-world data, right? This was published in a high-tier journal, Brain Stimulation, a clinic in Germany. Thank Andreas Buchner and their team. They were able to get the first 20 patients in Europe, and they published on this. They came back about 6 to 12 weeks. And you could see there that it was not as good, but comparable, I'd say roughly comparable to what we got in 10A2, where they had about, 10, 11 points, and we had, if I could see there, 13 points in 6 weeks. But I actually like the histogram plot there. You could see each bar is an individual patient, and you could see the distributions going left is improvement. It's not too different between our study and what happened in the real world. So I think that was encouraging. I think the most encouraging results is just what happened in the U.S. I have to give credit to Craig Casper. I don't know how he goes so fast and works so hard, but he already got about 50 patients in the short period of time through his clinic, and so he was the first to get most of this data that we can share. This was all done independent in his own clinic, so this is what's encouraging, and he was generous enough to present this last week and share it with us. But he got 47 patients through his clinic. 47 came back for the first follow-up. 23 he got coming back to the second, and they're also still coming in. That's as much data that he was able to get up until last week. He's been having good safety and ease of use with the treatment. In terms of the results, it was actually better than our 10 studies, which was encouraging. In the 47, within that first follow-up of roughly 6 weeks, they got 16.3 points in their THI. This was actually impressive. More than we got in our 10 studies was in the follow-up 2, they got to 24 points. They had 68.1% responders based on 7 points MCID at the 6-week point, and at the 12-week point, his clinic got 78% there with all the participants that have come in so far. Now, that's all the patients. If you actually break it up for what it's labeled for, which is higher moderate, above THI 38 and higher, they saw even better performance, which is consistent with what we saw in the clinical trials and FDA study, where you could see those who are less than 38 are only getting about 3 points on average improvement in their THI, and those that are greater than 38 are about 20.4. And then you see the percentage is up to 83% if it's greater than 38, and that was at 6 weeks, right? And then at 12 weeks, it goes up to about 90% of all his patients coming in. So we were quite encouraged. It's higher than I would have thought, but it's from our studies, so that was very encouraging. And so this is just summarizing what I said there. There's a minute left here, but key thing is that what we're finding from the real world is that continuing treatment is still driving further improvements in that moderate to severe group, and it is holding up consistently, both in terms of who it's labeled for, but also the results that we saw in 10A1, 2, and 10A3 studies. So that's encouraging. So all in all, there's a lot going on in bimodal space. You're going to hear other groups doing bimodal neuromodulation. So I believe it's here now, and there's different ways to approach it. Dr. Susan Shore is obviously working on face and neck stim. My lab, we're doing ear stim, but at the age, Linear has been paving the way and being able to demonstrate quite high percentage outcomes and consistency across clinical trials too. And I greatly apologize. I'm just the scientist. I'm not on the commercial team. So you may have questions of like, how do you get access to it? And I don't have the answer. But if you email me, then I can get you in touch with the right people. Because I am a professor, so I'm trying to stay away from that part of it. So with that, thank you very much for your time and for inviting me here.